Hello and welcome back. In this next video, we are going to be covering DNA replication at the replication bubble. So you should be using the two handouts that look like this, okay? And we'll be adding our annotations to these notes. All right. So the first thing which I want to indicate here is that what we're looking at here is the double helical structure of DNA. Now, before a cell can divide, we have to replicate that DNA. So there are enzymes which are going to open up the DNA, they're going to spread them apart, and they're going to create what is called a replication bubble. Okay, now inside of this replication bubble, we have exposed nitrogenous bases from these two strands, from these individual strands. So let's go ahead and give this a sequence just for kicks. All right, so we can say that we have A, G, C, T, A, C. All right, now what are going to be the nucleotides which are going to be across from these? But T, C, G, right? Okay. A, T, G. Right? So A to T, G to C, C to G, T to A to T, C to G. Now, what happens when we have these nitrogenous bases exposed, when we've opened up this replication bubble, is we're going to have the complementary nucleotides come in and pair with them. So T's will pair with A's, C's with G's, G's with C's, A's with T's, T's with A's, and G's with C's. All right? And then on the opposite strand, we would have A's coming in, A's, G, C, T, A, and C. So ultimately what is happening in this bubble is we get a complementary strand, complementary to the sequence of this exposed strand up here. All right? And then down on the bottom side. So these two lines in purple inside of the replication bubble, these are the new strands of DNA that are going to match the strands of this opened apart replication bubble. So notice what's happening. This bubble is getting wider and wider and wider. And then eventually what's going to happen is we're going to have a new double-stranded DNA composed of one of the original strands and one newly synthesized strand as shown here in purple. But not only that, but we will also have a second DNA molecule, all right, from this bottom original strand composed of the original strand and the newly synthesized DNA strand, all right? So that is DNA replication, like a big overview picture of how DNA is replicated, all right? But let's take a look at what occurs in one of these replication bubbles. So I'm going to zoom in. Let's say we'll zoom on this replication bubble right here and open this up. All right. So here is one big replication bubble. Okay. I'm going to use the color red because I think that's a little easier to see. Now, the enzymes which open up the bubble do so moving in opposite directions. And I'm going to indicate that little enzyme by drawing a little corkscrew. So, those are supposed to look like corkscrews that kind of look like sperm cells. But anyway, they're going to unwind the DNA in both directions. And these enzymes are named helicase. And helicase unwinds a double-stranded DNA double helix. All right? So its name indicates what function it performs. Now at the center of this replication bubble is where they began. This location is called the origin of replication. Now, once these helicase molecules have moved in opposite directions away from the origin of replication, we're going to expose these two strands of DNA on either side. So a second enzyme comes in called DNA polymerase, which is going to lay down new DNA in the 5', so this is the 5' end of this new strand, into the 3' end. All right? So going into the replication fork, this new strand is referred to as the leading strand. And all DNA polymerase has to do is just follow DNA helicase, and as long as DNA helicase is opening up more double-stranded DNA, DNA polymerase can follow in its wake to make the leading strand, which is synthesized continuously. Now, remember that DNA, double-stranded DNA, is anti-parallel? Well, one of the interesting things about DNA polymerase is that it can only synthesize new DNA in the 5' to 3' direction. So, can DNA polymerase go on this opposite strand and make new DNA in this direction? No. But a DNA polymerase molecule can land here and synthesize a little bit of DNA in the 5' to 3' direction. And then the DNA polymerase can come off and go around, and wherever DNA helicase exposes more single-stranded DNA, DNA polymerase can lay down another little piece in the 5' to 3' direction. So notice here that on the leading strand, this DNA is synthesized continuously, but on this side, this is the lagging strand, and it is synthesized discontinuously. All right, so we have identified two enzymes, helicase, which unwinds the DNA, DNA polymerase, which lays down the new strands, the leading and the lagging strand, and then there is a third enzyme called DNA ligase, which I will draw like glue, all right? So this is DNA ligase, and it will come in, and it will seal the backbone between these little pieces of the lagging strand. So ligase seals the gaps. All right? So I'm trying to keep this as simple as possible. I want you guys to understand the job of helicase, the job of DNA polymerase, the difference between the leading strand and the lagging strand, and the job of DNA ligase. And the information for this process, for this concept, comes from chapter 10. So I'm bringing in a little bit of outside information from another chapter into this chapter so that we can understand the mechanism of DNA replication at the replication fork. Okay, so until next time, see you guys soon.